In today's video, we're going to go through the process of Monstera Albo tissue culture. You might think, well, Ori, how are you going to do that when Monstera Albo is not generally tissue cultured on scale? Because it's not genetic. The Monstera Albo is derived from a chimeric mutation, so the variegation is not hereditary, not genetic. It's not the same for every cell of the plant. Well, yes, but it's still tissue cultured, it still works. It's just that from your explant, when it multiplies and forms calyces, the certain part of a clones will be entirely white, which will not survive under a general conditions, and the certain part of our elbow will be entirely green, which is not really what we want. So yeah, let's go through the process of a media preparation, a sterilization of our flasks, and yeah, let's go through the video. Hope you enjoy it and find it somewhat helpful. So first of all, this fucker is an air sterile still box in which many people actually preach you to do tissue culture, but I found that it's very troublesome. So it's gonna cost you more than you would anticipate to make from essentially from probably selling the plants. So do not try this shithole. It's not worth it. It's the worst thing that I've done in this lab. But what you can actually do is get a laminar flow hood. This is the angel dropped from heavens on earth. It's like the general purpose of a laminar flow hood is to provide us with a sterile airflow so we do not get our media and plant contaminated. This is a very simple device. I've made it myself with a couple of fans, a functional sterile HEPA filter, and a power source. Basically, this is one from an old computer device. It's very easy to make and very cost effective, and this fucker will cost you more than this one. So I suggest you strongly to just get a functional HEPA filter and build yourself a functional laminar flow hood. It's a very simple device, nothing very complicated. And now, by the mighty power of science gods, let us begin the salts preparation. The preparation of our media. Here's our 250 milliliter flask. Here's our agar. And where is it? Here's our MS media, basically all micro and macronutrients for our plants to thrive in. It's quite simple. When people say that tissue culture is a complicated process, it's not. It's the complicated part is maintaining the sterility necessary for our plants to not die. That's the complicated part. Anything other than that is child's play. Here I'm adding appropriate amount of MS cook, which is micro and macro elements for our plants, and agar and sucrose, which is essentially cane sugar. In my case, I decided to make 100 milliliters of media, which is ample for my plant. Plant growth regulators, PGRs, are really important. Here I'm adding 7.5 milligrams per liter of BAP and 0.5 milligrams per liter of NAA. pH level for Monstera Albo and Monstera Tycoon is 5.7. We take our pH meter, turn it on. And we're gonna wait for it to finish reading. So we can say that it's from between 5.2 but it still goes down.
So just because our pH level is slightly lower than required, we're using pH buffers, aka certain chemicals which can rise or lower pH levels. In my case, I need 5.7 to 5.6.
Do you know what is the most frustrating part of a biologist's life? It is this: a broken flasks and beakers. This is a the Great Depression.
So here we are sterilizing our explants into a 10% bleach with a dash of dish soap. Dish soaps were important to break the surface, and after 25 minutes we'll have an explant which is quite sterile on the surface at least, and will be quite ready to move to a multiplication media. Keep in mind that everything put into a laminar flow hood has been prior sterilized. Scalpels, jars, stir bar, even the containers which we use for sterilization obviously have been sterilized a priori. It is very important because without sterilization, the point of laminar flow hood is wasted. After those 25 minutes, we move our explants into a sterile distilled water to remove the residual bleach and dish soap to clean the explants, and we put our explants into a sterile petri dish to cut off the ants, because ants have been very much soaked with a bleach, so they are kind of dead. Well, that's basically it. We take our sterilized explants and put those tossers into our multiplication media to multiply and grow. That's it. Cleanliness is the key in tissue culture. It's a very simple process, really. You just have to always be clean, always work under a sterile hood, and always sterilize the caps whenever you do a transplant or any sort of work like this. It's quite essential because even one microbe which might be dropped into your flask will multiply exponentially and ruin your clones, your explants, very unfortunately, and your work will be ruined. So always work very cleanly, very safely, and your plants will be alright.
So these babies in my flask now will be placed under a full spectrum lighting system and will be monitored for a week to find out if the flask is contaminated. Well, hopefully not, but everything happens even under the laminar flow hood, which is a sterile environment by a book. It's not 100% guaranteed that the flask will be 100% sterile because of airborne contamination, unsterilized flasks, unsterilized scalpels, and more stuff like that. There are a lot of variables in tissue culture, but hopefully we will see some callus formation in about a week because the cytokinin concentration here is quite high. It's 7.5 milligrams per liter, which is abnormally high for houseplants and tropical plants. And yeah, hope you guys enjoy this video. I will see you in about a week to do the update. And you can also follow us on Instagram. It's called Synthiogenesis. It'll be somewhere over here. And see you next time. Bye.